Concepts of surgery. The most simple way to learn surgery. Do you know when was the first successful treatment of cancer by immunotherapy used? Between 2000 to 2024. Between 1950 to 2000. Between 1900 to 1950. Between 1850 to 1900. Answer is 1891. In 1891, William B. Coley, a bone tumor surgeon, injected streptococcal organisms into a patient with inoperable cancer. He thought that the infection he produced would have the side effect of shrinking the malignant tumor. He was successful, and this was one of the first examples of immunotherapy. This idea came from his observation on a patient named Fred Stein. He had advanced sarcoma neck. While on treatment, he developed erysipelas, and his sarcoma regressed and disappeared after developing infection. Over the next 40 years, as head of the bone tumor service at Memorial Hospital in New York, Coley injected more than 1,000 cancer patients with bacteria or bacterial products, inciting the immune system. These products were named Coley's toxins. He and other doctors who used them reported excellent results in some cancers. Despite his reported good results, Coley's toxins came under a great deal of criticism. However, the modern science of immunology has shown that Coley's principles were correct and that some cancers are sensitive to an enhanced immune system. William B. Coley, a bone sarcoma surgeon, deserves the title Father of Immunotherapy. Often it is found by pathologists examining histology of tumors that tumors often have lymphocyte infiltration between cancer cells. These lymphocytes are called tumor-infiltrating lymphocytes. These are mainly CD8+ lymphocytes. So what is the importance of presence of lymphocytes inside tumor? Those tumors, who have strong presence of tumor-infiltrating lymphocytes, have better prognosis. So if immune system is strong, then how does tumor cells escape clutches of our immune system to develop a full-fledged tumor? Let's see the mechanism of immune escape, understanding which is crucial in development of immunotherapy. There are 8 important mechanisms of immune escape. I have given them a title for ease of remembrance. 1. Invisibility. Since recognition by T lymphocytes involves the binding of their antigen receptor to the MHC complexes, cells that express low or no MHC will be poorly recognized and listed by cytotoxic T cells. In human tumors, complete MHC class I loss is frequent, and occurs in many types of cancer. 2. Early satiety. Cells in the body losing expression of MHC class I molecules can be detected by NK cells, which are innate lymphoid cells. NK cells have NKG2D receptors on their surface. Ligand for this receptor is called MICAB and expressed in cells under stress. This ligand binding alerts NK cell and it attacks the cells expressing these ligands. Many tumors lose the expression of these ligands, or even shed them in soluble form, which bind to receptors on NK cells and saturate them to induce a downregulation of NKG2D at the surface of NK cells even before they reach the tumor cells. By the time they reach cell they are no longer hungry. 3. Conversion. Tumors have undergone several mutations leading to expression of new altered antigens on its surface. This altered cell is recognized by immune cells as foreign cell and attacked. Macrophages infiltrate tumor and start attacking tumor cells. But tumor cells secrete a cytokine which converts macrophages in another form called M2 macrophage. M2 macrophage has a property of secreting chemicals which are angiogenic and help in tumor growth by suppressing lymphocytes. So tumor has converted our own cell and now using it for resources and against our immune system. 4th. Recruitment. Next, tumor wants more such M2 macrophages. So it secretes chemokines CCL2 and colony stimulating factor 1, which attracts more macrophages and converts them to M2 macrophages. In way macrophages are lured in and converted. 5th. Exhaustion. Tumors may also evade antitumor immunity through the secretion of immunosuppressive cytokines and factors, like transforming growth factor Greek letter beta, TGF-beta, and interleukin-10, which deactivate the immune response. 6th. Deep attack. Lymphocytes need supply of crucial amino acid tryptophan for function. Tumors are able to deactivate the cytotoxic T cell responses by promoting the degradation of the crucial amino acid tryptophan through the overexpression of enzyme indolamine 2,3-deoxygenase. This enzyme catalyzes the conversion of tryptophan to kynurenine. The depletion of tryptophan and the production of kynurenine induces of lymphocyte cell apoptosis. 7th. Headquarter. Tumors may have ability to recruit myeloid cells such as monocytes and immature precursors from the bone marrow, which are transformed into polarized M2 macrophages. Inflammation, although, is vital for protecting body, but if it is not controlled, then it can spread to whole body and become dangerous. There is delicate balance between pro-inflammatory and anti-inflammatory process. When work is over, anti-inflammatory process supersedes and inflammation ends. There are certain receptors on lymphocytes which, when activated by ligands, suppress lymphocytes. These ligands are produced by other inflammatory cells, like programmed death-1 and CTLA4. These are appropriately called immune checkpoint receptors. Tumor cells activate these receptors like PD-1 and CTLA4 by producing their ligands, and hence suppresses inflammation. Protecting themselves, tumors are able to express immunosuppressive ligands, such as PD-L1, which, when bound to their receptor on the activated T cells, promotes their apoptosis. So what is immunotherapy? Immunotherapy is ability to recruit more lymphocytes and train them in identifying and kill tumor cells. There are mainly four mechanisms in which immunotherapy has been used. For an antibody to target antigen, antigen must be present in majority of tumor cells and should be specific to tumor cells to minimize collateral damage. Once such antigen is identified, antibody against it can be used to target tumor cell. Like epidermal growth factor receptor expression on colorectal cancer led to use of anti-EGFR antibody, cetuximab. Expression of HER2 protein on breast cancer cells led to use of anti-HER2 therapy, trastuzumab. Bispecific antibodies have the advantage that they can either simultaneously target two antigens and thus increase the specificity of the antibody to the tumor, and reduce the non-specific toxicity, or one antigen can target cancer cell and another can attract immune cells, and so bring immune cell closer to tumor cell. Like blinatumomab, it is composed of two antibody fragments, one targeting CD19, and the other one targeting CD3. CD19 binds to malignant B cells which express CD19, and CD3 binds to cytotoxic lymphocytes which express CD3, thus bringing immune cell and tumor cell closer. FDA-approved blinatumomab is used as treatment for refractory B-all, Philadelphia chromosome negative, and it increased the chances of complete response in these patients. 2. Immunomodulatory drugs. Immunomodulatory drugs work in two ways. They either block immunosuppressive receptors, like PD-1, so that tumor cells are not able to suppress lymphocytes by activation immunosuppressive receptors, or they activate stimulatory receptors which make lymphocytes more agile and effective. Hence anti-CTLA4 and anti-PD-1 antibodies block these receptors and prevent apoptosis of lymphocytes plus stimulates lymphocytes. A fully humanized anti-CTLA4 called ipilimumab, and anti-PD-1 antibody nivolumab and pembrolizumab. Cytokines. It was in the late 70s that the ability of cytokines to mediate lymphocyte survival and proliferation was discovered. This new development allowed researchers to grow T and B as well as innate immune cells in culture for in vitro studies. Following this, recombinant cytokines were developed. 2. Cytokines have mainly been tried in systemic cancer treatment. Interleukin-2 is a cytokine promoting lymphocyte survival and proliferation, which explains the rationale behind its use in cancer therapy. Systemic administration of interleukin-2 is a non-specific treatment, aiming at boosting the proliferation of NK and T cells. They were initially tried in melanoma and kidney cancer. They have significant side effects like diarrhea and hypertension. Another cytokine that drew interest for immunotherapy was interleukin-12. Interleukin-12 is produced by activated dendritic cells and induces the activation of NK cells as well as helps the differentiation of helper T cells into pro-inflammatory form. However, treatment with interleukin-12 was found to have strong adverse effects and limiting its use. Cancer vaccines. Cancer vaccines are of two categories: prophylactic vaccines and therapeutic vaccines. Their prophylactic activity is generally mediated by the induction of stable, long-lived high levels of neutralizing IgG or IgA antibody responses. Such prophylactic vaccines cannot be applied to most cancers, with the exception of virally induced cancers. For example, the human papillomavirus is a known trigger for cervical neoplasia. Vaccines against HPV have been developed and could successfully prevent HPV infection as well as neoplasia development. In contrast to prophylactic vaccines, therapeutic cancer vaccines may protect the host from tumor growth not by neutralizing antibodies, but through the induction of tumor antigen-specific T cell responses. Peptide and adjuvant-based vaccines aim at igniting an adaptive immune response against a chosen target tumor antigen. Antigens of highest interest for therapeutic vaccines include mutated antigens, neoantigens, that are overexpressed in the tumor compared to normal tissue. Finally, tissue-specific antigens can be considered when the tissue happens to be dispensable for the survival of the patient. For example, the prostate-specific membrane antigen PSMA, or the B-cell antigen CD19. Naked DNA and recombinant vector-based vaccines. The delivery of tumor antigen via its genetic sequence has also been studied. Bacterial plasmids modified to include the gene of the antigen of interest can be directly injected into the muscles of patients, and some cells, such as dendritic cells, will uptake the genetic material and start expressing the protein. It can then be presented to T cells via the classical MHC class I pathway of antigen processing and presentation. To circumvent the hurdle of the antigen's identification and selection, researchers have explored the use of whole tumor cell vaccines. Lizards of autologous tumors provide the whole spectrum of tumor antigen and neoepitopes, thus allowing to skip the identification of target antigens. A fourth modality of cancer immunotherapy is the adoptive cell transfer. Tumors are often infiltrated with T lymphocytes. However, due to the chronic antigen exposure as well as the immunosuppressive environment, these T cells are functionally impaired. Researchers sought to isolate those TILs and expand them in vitro, in the presence of high interleukin-2 levels, aiming to artificially increase their total number and to reactivate them away from the deleterious cancer environment. Subscribe to our YouTube channel to learn surgery in simple way.